Hello, this is SabahMusfiah. Today I'll present a short summary of the paper titled Dynamic Growth Monitoring of Saccharomyces cerevisiae based on microwave sensor array. Let's begin. In this paper, the researchers designed a microwave sensor array to monitor yeast growth in real time without any physical contact. They used 9 resonators and analyzed multiple microwave parameters to track how the yeast grows and changes over time. One key finding is that a combined parameter called MSP3 showed very high accuracy, with an R-square value of 0.9985, making it a strong indicator for evaluating yeast growth and cell behavior. Saccharomyces cerevisiae, especially the YPH499 strain, is widely used in the brewing industry. Monitoring its growth in real time is important for maintaining flavor, yield, and overall product quality. Most real-time monitoring methods only work in liquid cultures, but solid agar is more useful for observing colony patterns. So a non-contact method for monitoring yeast on solid media is needed. Microwave sensors are suitable because they allow sensitive, real-time, and non-contact detection. Previous studies also showed that microwave resonators can monitor bacteria and yeast. Since microwave sensing generates many parameters, combining them improves accuracy. Methods like PCA and Dempster-Shafer theory help fuse these parameters. In this study, the authors used a nested split ring resonator sensor array with 9 resonators. By combining the data using PC and DS theory, they created a synthetic parameter called MSP3, which strongly correlates with yeast survival and apoptosis. In this study, the authors designed a nested split ring resonator array, which is a modified version of the traditional SRR. Each resonator behaves like an LC circuit, and by changing the number of nested rings, the resonance frequency can be tuned. The array contains 9 resonators, each producing a different frequency, and simulations confirm that the gap region provides the strongest electric field for sensing. For microwave monitoring, 4 parameters are measured: 2 resonance-based and 2 non-resonance parameters. PCA was used to extract the most informative signal, and then Dempster-Shafer theory combined this into synthetic microwave parameters called MSPS. These MSPs are later linked to biological conditions such as cell survival or apoptosis. At the same time, yeast colonies were stained on solid agar using methylene blue to determine live and dead cells. Microscopy was used to count total live and dead cells at each growth stage. From these, the survival and apoptosis rates are calculated and mapped to the MCPS to validate the microwave sensing results. Here, yeast growth affects the microbe response in several ways. As the colonies grow through different stages, the dielectric properties around the resonator changes. This leads to shifts in resonant frequency and variations in insertion loss. Non-resonance parameters like FSI and FWHM follow the biological growth pattern more smoothly. They track slow growth, rapid growth, the plateau, and the final decline very reliably. These parameters are more stable, making them better indicators. All of these microwave parameters are lifted to a logistic S-shaped growth curve, while the fit varies across the 9 resonators. Overall, the non-resonance parameters show the best match to the yeast growth trend. PCA was used to combine data from all 9 resonators. The first principal component captures most of the meaningful information, including almost all of the non-resonance parameter variation. This shows that combining resonator data improves the growth trend prediction. From the staining experiments, we measured total cell number, survival rate, and apoptosis rate. As yeast grows, total cells increase, while survival decreases and apoptosis increases, which is consistent with typical microbial growth behavior. Finally, Dempster-Shafer theory was used to fuse the microparameters into nine synthetic parameters called MCMSPs. Among these, MSP3 showed the strongest correlation with survival and apoptosis. It achieved a very high accuracy with an R-square of 0.9985. In conclusion, this study developed a microbe monitoring system using a nested split ring resonator array to track yeast growth in real time. Using all nine resonators together provides much better performance than relying on a single resonator. By fusing multiple microparameters, the authors created a synthetic parameter called MSP3, which showed a very strong correlation with yeast survival and apoptosis. This makes MSP3 a reliable indicator for evaluating the brewing process. For future work, the researchers plan to test additional microfrequency bands and develop improved device structures that can be used for large-scale brewing applications. Thank you. That's it for today.